Dear students, in this session we will be discussing about apoptosis or programmed cell death. So, what does the word apoptosis means? It means falling off in Greek. And what is apoptosis? It is a process that occurs in multicellular organisms for the greater good of the whole organism. Here, a cell intentionally decides and commits to die. This often occurs when the cell's DNA is damaged and there is a possibility that it may become cancerous. Apoptosis is also known as programmed cell death because it is biochemically programmed with clear instructions in the cell's DNA. It happens without causing any damage to the nearby cells or the whole organism. There is another method of cell damage termed necrosis, which is highly destructive. Necrosis occurs when a cell dies due to trauma or nutrient deprivation. So apoptosis is termed as programmed cell death, which happens without causing any damage to the nearby cells. There are two major types of apoptosis pathways, and they are intrinsic pathway and extrinsic pathway. In intrinsic pathway, a cell receives a signal to destroy itself from one of its own genes or proteins due to any DNA damage or any other internal cellular stress. And in extrinsic pathway, a cell receives a signal to start apoptosis from other cells in the organism, from the exterior of the cell. The extrinsic pathway may be triggered when the organism recognizes that a cell has outlived its usefulness or it is no longer a good investment for the organism to support. So there are two apoptosis pathways, the intrinsic pathway and the extrinsic pathway. So when does apoptosis of a cell occur? A cell undergo apoptosis when its existence, the existence of that particular cell, is no longer useful to the organism. For example, a cell that cannot function properly may initiate apoptosis. When the cell is experiencing stress due to the deprivation of oxygen, the cell itself decides that it is dangerous or costly to the host organism, and it will commit apoptosis. Or if a cell has become badly stressed or damaged, it may commit apoptosis to prevent itself from becoming dangerous to the organism as a whole. Cells with DNA damage, for example, may become cancerous, so it is better for them to commit apoptosis before becoming cancerous. In the third scenario, the cells may commit apoptosis because the organism does not need them anymore due to the natural development of the organism. One famous example is that of the tadpole, its development into frog, when its gill, fin and tail cells commit apoptosis as the tadpole metamorphoses into a frog. These structures, gill, fin, tail, etc., are needed only when the tadpole lives in the water. It is no more needed as the tadpole metamorphoses into a frog. So these are the situations when apoptosis occur. So what are the functions of apoptosis? In human beings, apoptosis plays a major role in preventing cancer by causing the cells having DNA damage to commit suicide, to commit apoptosis before they can become cancerous. Apoptosis destroys precancerous cells and cells that are no longer useful to the organism. It also plays a role in the atrophy of muscles, where the body decides that it is no longer a good idea to spend calories on maintaining the muscle cells which are not being regularly used. Apoptosis is also required for embryogenesis, metamorphosis, endocrine dependent tissue atrophy, normal tissue turnover, that regularly happen in the gut tissues, etc. Apoptosis is also reported in a variety of pathological conditions. So, let us discuss what is apoptosis and necrosis. So, necrosis is a pathological condition where the cells die due to acute injury or stress. During necrosis, the cells will swell and burst, and during this process, they will spill their contents, and the lytic enzymes that get released from the cell into the exterior will result in a damaging inflammatory response, thereby destroying more and more neighboring cells. While in apoptosis, this is a neat process whereby the cells dies neatly without causing any damage to the neighboring cells. Here, the cell which is undergoing apoptosis will shrink and condense. Their cytoskeleton will collapse, nuclear envelope will disassemble, nuclear DNA breaks up, small apoptotic bodies are formed, and these apoptotic bodies are rapidly phagocytosed by phagocytic cells. So, in apoptosis, the contents are recycled and there is no inflammatory response in apoptosis. So, as we discussed, in necrosis, cells die as a result of acute injury. They swell and burst, spill their contents all over their neighbors, and cause a potentially damaging inflammatory response. Their organelles are not functional during necrosis. While in apoptosis, the cell dies neatly without damaging the neighbors. The cell shrinks and condenses. The nuclear envelope disassembles. Nuclear DNA breaks up into fragments. The cell forms small blobs known as apoptotic bodies, which are rapidly phagocytosed. And so there is no leakage of the contents during apoptosis. As a result, there are no damaging consequences during apoptosis, and the organic components of the dead cells will be recycled. Now, we will discuss the major cytological changes that happen to a cell during apoptosis. The cell will shrink in size, its size will be decreased, the cell will undergo fragmentation, cytoskeleton will collapse, the nuclear envelope disassembles, the cell will release apoptotic bodies, small membrane-bound bodies, and these membrane-enclosed cell fragments or the apoptotic bodies are rapidly phagocytosed by macrophages and other cells. These events can be observed from the image as well. A normal cell during its apoptosis will decrease in its size. There will be cell shrinkage, chromatin condensation, membrane blubbing, the organelles will disintegrate, the nucleus and organelles will collapse, and they will get arranged into membrane-bound apoptotic bodies, and these apoptotic bodies are phagocytosed by phagocytic cells. So these are the major cytological changes of apoptosis. Now we will discuss the mediators of apoptosis. They are termed as caspases. The intracellular machinery responsible for apoptosis depends on a family of proteases. They are known as caspases. They have a cysteine at their active site and cleave their target proteins at specific aspartic acids. So they are termed as cysteine aspartic proteases or cysteine aspartases or cysteine dependent aspartate directed proteases. In short, they are termed as caspases. These apoptotic caspases are subcategorized into two: initiator caspases and executioner caspases. Examples for initiator caspases are caspase 2, caspase 8, caspase 9 and caspase 10, while caspase 3, 6 and 7 are executioner caspases. These caspases are synthesized in the cell as inactive precursors or procaspases. These procaspases are usually activated by cleavage at aspartic acids by other caspases. Once activated from the procaspase into an active caspase, they cleave and activate other procaspases, resulting in an amplifying proteolytic cascade. The initiator caspases autoproteolytically undergo cleaving and activation, while the executioner caspases are cleaved by the action of initiator caspases. Once the initiator caspases are activated, they produce a chain reaction, activating several other executioner caspases. These executioner caspases are capable of degrading over 600 cellular components, and they induce the morphological changes for apoptosis. For example, some of the activated caspases cleave other key proteins in the cell. Some cleave the nuclear lamins, causing the breakdown of nuclear lamina. Some other caspases cleave a protein that normally holds a DNA degrading enzyme, thereby activating or freeing the DNase, which will cut up the DNA in the cell nucleus. In this way, caspases cause the dismantling of the cell in a quick and neat way, help to form small blobs known as apoptotic bodies, which are rapidly taken up and digested by phagocytic cells. So, here we can see the procaspase activation. As we discussed, caspases are synthesized as inactive precursors or procaspases. They are activated by cleavage at aspartic acid residue by other caspases, and they become active caspases. And then comes the proteolytic caspase cascade, where the activated caspases cleave and thereby activate other procaspases, resulting in an amplifying proteolytic cascade. This activation of the intracellular death pathway, the triggering of the proteolytic cascade, is usually triggered in a complete fashion, all or none fashion. The proteolytic cascade is not only destructive, it is also self-amplifying, it is reversible, so that once a cell reaches a critical point along the path to destruction, it cannot turn back. Thus, after this proteolytic cascade, as we discussed in the previous slide, these activated caspases cleave key proteins such as nuclear lamins. They activate DNA degrading enzymes, DNase, and enables the self-dismantling of the cell towards apoptosis. So, there are two major types of apoptosis pathways, the extrinsic pathway and the intrinsic pathway. The extrinsic pathway of apoptosis begins outside the cell when conditions in the extracellular environment determines that a cell must die, a cell must undergo apoptosis. The intrinsic pathway of apoptosis pathway happens when injury occurs within the cell and the resulting stress activates the apoptotic pathway. In both the intrinsic and extrinsic pathway of apoptosis, signaling results in the activation of caspases that act in a proteolytic cascade for initiating apoptosis. Now, we will discuss about extrinsic pathway. It is also known as the death receptor pathway. This is initiated by the activation of death receptors on the cell surface. For example, killer lymphocytes can induce apoptosis by producing a protein known as FAS ligand, which binds to the death receptor protein FAS or FAS receptor on the surface of the target cell. The FASR proteins will cluster, and the clustering of FASR proteins activates the death domains at the cytoplasmic tail of the receptor. It will activate the adapter protein FADD, which will further recruit procaspase 8. FASR, FADD and procaspase 8 together will form the death-inducing signaling complex or DISC. The procaspase 8 molecules will cleave and activate one another, and the activation of procaspase 8 to active caspase 8 will either lead to the downstream activation of the intrinsic pathway by inducing mitochondrial stress, or it may lead to direct activation of executioner caspases and apoptosis. The image shows the extrinsic pathway. Here we can see the interaction between FAS ligand from the killer lymphocyte with FAS protein receptor on the surface of the target cell. This results in the recruitment of intracellular adapter proteins, which will result in the aggregation of procaspase 8 molecules. The procaspase 8 molecules will cleave and activate one another and will become active caspase 8 molecules. Then they can activate the downstream procaspases to induce apoptosis. From this point onwards, there are two possibilities. The active caspase 8 molecules can either lead to the activation of intrinsic pathway by inducing mitochondrial stress and the release of cytochrome C from mitochondria, or these active caspase 8 molecules can directly lead to apoptosis by activating the executioner caspases. The second pathway is intrinsic pathway. It is also known as mitochondrial pathway. Intrinsic stresses such as arising from oncogenes, direct DNA damage, hypoxia or less oxygen, survival factor deprivation, etc. can activate the intrinsic apoptotic pathway. P53 is a sensor of cellular stress and is a critical activator of the intrinsic pathway. P53 initiates apoptosis by the transcriptional activation of pro-apoptotic BCL family members, and also P53 inhibit the anti-apoptotic BCL2 proteins. P53 also activates other genes which are contributing to apoptosis and genes that lead to increases in reactive oxygen species. The reactive oxygen species or ROS lead to oxidative damage to the mitochondria. As a result, the mitochondria are induced to release the electron carrier protein cytochrome C into the cytosol. Cytochrome C will bind to an adapter protein known as APAF1, which recruits the initiator procaspase 9 molecules. This leads to the formation of a caspase activating multi-protein complex called the apoptosome. The procaspase 9 will get activated to active caspase 9, which will cleave and activate other executioner caspases and leads to the degradation of cellular components for apoptosis. So what is apoptosome? The apoptosome is a large quaternary protein structure formed during the process of apoptosis. It is a multi-molecular hollow enzyme complex assembled around the adapter protein APAF1. Its formation is triggered by the release of cytochrome C from mitochondria. The apoptosome triggers the activation of caspases in the intrinsic pathway of apoptosis. Once activated, this initiator caspase can then activate effector caspases and trigger a cascade of events leading to apoptosis. This image shows intrinsic pathway or mitochondrial pathway. Here, during intrinsic pathway, any damage to the cell will result in the release of electron carrier protein cytochrome C from the mitochondria into the cytoplasm. This released cytochrome C will bind and activates the adapter protein known as APAF1. APAF1 binds and activates procaspase 9. The activated procaspase 9 activate downstream procaspases to induce apoptosis. This image shows both extrinsic and intrinsic pathways of apoptosis. So as we discussed, the external stimuli activate death receptors in the extrinsic pathway, resulting in the formation of activated caspase 8. In intrinsic pathway, the cytochrome C release results in the activation of caspase 9. The active caspase 8 or caspase 9 further activates procaspase 3 to caspase 3, which further mediates the apoptosis. The executional caspases such as caspase 3, 6 and 7 can degrade over 600 cellular components, and they are the main mediators of apoptosis as we discussed in a previous slide. Executional caspases are termed so. They are termed as executioners because they coordinate the destruction of cellular structures such as DNA fragmentation or degradation of cytoskeletal proteins. So, we discussed about apoptosis and necrosis, about caspases and the pathways of apoptosis, the intrinsic and extrinsic pathways of apoptosis. Thank you so much for listening.